Welcome back for our concluding session for our second keynote. And I think you would all agree what a fantastic 2 days it's been. And I'm very excited for our final talk today. So I'm very pleased to introduce Per Bork, who received his PhD in biochemistry and then in Leipzig University, and then continued with his habilitation in theoretical biophysics. And got that in 1995 from the Humboldt University in Berlin, and started at EMBL after that, and is now the director of EMBL in Heidelberg. And I want to mention two kind of notable accolades, which I just learned about, one of which was he was awarded the Novozymes Prize, which I thought was, what is this? And it was for developing groundbreaking, publicly available and integrative bioinformatics tools by the Novo Nordisk Foundation. And he was also awarded the 2021 International Society for Computer Biology Accomplishments by a senior scientist for his contributions to the field. So I'm delighted to invite him up here to tell us about everything that's been going on and the look towards the future.
Yeah, so thanks a lot for the kind introduction. Thanks a lot for having me here. So the purpose today, how I see it for my talk, is not so much talking about algorithms, but how to apply that, where can things go, and with my usual modesty. So we are a bioinformatics group, and what you can see here, sort of the pillars of our work. So really what we're up to for quite a few years already, but it's not so prominent. We have the modest idea of really understanding molecular fluxes of function in our planet. So, but this is step by step to be approached. So we worked for many years still working actively on a human gut microbiome. And I see the human gut as a kind of warm-up because it's rather simple from a bacterial point of view community. It's pretty well studied. It's a huge research funding went into this, and there's access to experimental information. So you can put quite a few things in a lab, and I come to this, which is, I think, for all these things, if you go out to the world, very much needed. What's also needed, obviously, is in a computational infrastructure, and we have a lot of it. Actually, we do a lot of algorithmic discussions as well. But when I go straight to the talk, which is a structural thing, I will just briefly touch on the infrastructure part, more from a conceptual point of view, and that goes beyond metagenomics. Then I will give you some ideas on the human gut microbiome, where some challenges are there. And then I go out to the planet, where we're still in a warm-up phase, if you wish. And along the way, I try to highlight challenges in red, which are more computational of various source, from algorithms to storage and whatever. So let's start just with a concept on infrastructure. So here on the campus, there's also EBI, also Sanger Center, there's huge infrastructure, huge data resources, but there's a whole layer of groups on resources that's just one step below. And EBI is 800 people, but still they have to select very carefully what they take on, what they develop, etc. But there's huge word out there, how to do it. As a normal PI, you have a cool tool, then the PhD student runs away and either dies or the student is smart enough to take it. So, but over time, and I'm an oldie, how to develop this? So our concept that we excellently started, and I think I'm really a fan of it now, was really giving the tool, letting it go if you develop in a group to the people who have a heart on it, but stay in connection, make the interoperability available, and build up the synergy, because two little tools together have much more power than individual one. So it started this was the situation 2006 you see here, and over the years things were developed in the group and spit out, but again developed and maintained in collaboration. And we have a situation now of a real network across the globe, if you wish, that really is a little layer of overhead. You have to communicate, but it has lots of potential. And we are also in a network of German infrastructure, and they develop lots of KPIs, when is a tool still useful, et cetera, et cetera. So what you can see here is these kind of stats that they're spreading over Germany. And the first thing you might see is that there's a correlation between usage and when tools were developed. So the late ones, they're still, they take them a while, they still might die, or they may still make it. So you need lots of persistency indeed to keep going in the field. Obviously, you have the right decision on what you develop, etc. So I think what we have now is a kind of federation of tools. It's a little more overhead than just one-to-one talks and just taking them in and communicating with the other one with another tool and these pipeline developments. But this concept mitigates various risks, from funding risks to start with. You can buffer a little, but also it allows fast evolution of tools, because each one evolves and still you keep the connection and remains very flexible. If you're too over-organized, you might sort of miss opportunities because you're stuck in your framework. It comes with issues, obviously, as well, like a very complex versioning on the data side, but also on tool side, and often it delays update cycles. I come back to this. I just want to throw it in for each PI in a way that might be a model that grows and amplifies the power of individual resources. So, but with that, I'm done with the kind of infrastructure part, and want to just give a background on the human gut microbiome and where the issues are also on different fronts. It started more than 10 years ago. I think the first big funding initiatives were 2008 or so. There was a HMP project and MetaHit in Europe, HMP in the US. But it was all about is finding associations between diseases and gut microbiome changes. And it's like epidemiology. It's the same kind of principles and the same kind of issues. And as soon as it started, really, there were tons of associations being published, mostly on 16S, a small marker gene, but more and more coming the proper metagenomic approach, because it's getting cheap enough to scale. Still, we have to say, after all these years, and now it's almost every week a new association, we don't know really what healthy gut microbiome is, and we have to note that these associations can be very unspecific. Because you have only a few angles into the gut microbiome, there are lots of factors that needs to be considered. Just to give you the classical route, what the idea was based on, how it takes time-wise as well, if you want to develop it, because why do you do all these associations? At the end, you want to have a diagnostic tool. And that's another layer, quite often out of the academic box. So we took on this part early on. For colon cancer, this was really early days, and based on 150 samples, and we found some species markers that nicely were specific for early stages of colon cancer, not for adenomas, so pre-stages. But obviously, there were tools out, so stool tests were out already, so all the point is not being non-invasive here. And at that time, we were better than a tool that was out there, so like fecal cut blood test. But the year after we published, several European countries took the FIT test on, which was a little better. So also on that front, a battle if you want to go and make it to diagnostics. Anyway, the good thing was that the microbial readout was very different from the blood in a stool readout. So if you combine the two, really had a huge leap with medical implications. That was a nice idea, 2014. But then metagenomics comes in, and these samples were from Europe. So you have to make sure that you don't have a specific city effect or whatever. So you have to do meta-analysis. And we did only in 2019, 2 different groups. Also Nicolas Agata. It's very similar concept, but approach is very different. Different data processing, different data sets, etc. We come to the same conclusion. It seems to be spread also in Asia. We didn't have data in Africa, so there's also this developing country kind of living style component sort of built in. But at least it was a big thing. So this is cool. We have now something based on 20 species, nice biomarkers. But if you do metagenomics, even if prices go down these days, it's probably still 200 euros plus a profit the company wants to make. So it's a no-go if you think in economic terms. So what we did then here, the labs comes in as well. We took just primer-based, very classical qPCR approaches. So finally, we managed to have something that's actually even higher quality. Are now pushing it with a large diagnostics company. Unfortunately, due to COVID, it takes a little longer. They make much more profit on the COVID front. But it looks like it has a chance, really realistic, going into the market. So the good thing is, your first time is always the hardest. We did the similar things for pancreatic cancer much more recently, and so the timelines are much shorter. But here we have different issues because it was kind of low biomass, so very few species, and you're prone to contamination. And this comes to a challenge in all of these things. Independent of the low biomass is a very specific challenge in a tissue, and there's big debate in the community, why and why not. And a good arguments in both directions. I don't want to step in there. But in any case, you can't show, with all the approaches so far, specificity. So we have by now 6,000, in that case we had 6,000 other studies with disease associations, etc. We could take the control group, could look with comorbidities with other diseases. There were a couple, but there might be 50,000 others out that we could not test. You know, that's always an issue. That means you have the huge effort to preparing for clinical trials. So these are things which are really hard work, much harder than expected, of course, but it's at the end, it still looks very good that the microbes really can contribute. Despite there are lots of small companies, but really the rolling out of microbiota in the gut for diagnostics hasn't really arrived yet, despite all the time and hype. So, but the big bug is any of these associations, again, like in human, in these associated studies, there are lots of confounding factors. And many of these interactions you see are very indirect, and we came across quite a few cute cases. I don't go into this today, and just give you an overview, you know, what the known factors are. And so on that circle, you know, even the gut microbiome is not homogeneous. They're age-dependent stage, so infants look very different from adults, for example. Their composition is stage, I come to this in a moment, and there are lots of stochastic effects, environment effects. In Asia, things look different, or different parts of Asia even. There are lifestyle effects, and it's a genetic component. So over the last, 10 years or so, I think we studied whatever we could and figure out individual factors, you know, which factor is influencing most. Others studied the genetics. Aaron Segal, for example, had a nice paper saying that host genetics is in the order of 2% only contribution, but that doesn't say that a single SNP can have a very stellar kind of association with a very particular disease. It's just averaging. And so after, I want to go in now into a kind of algorithmic issue, and that's the enterotypes, one of these factors. So we stumbled across it early on, already 2011, when the first data came out, and that was a clustering based on 39 samples. We had about 60 S data sets with more, etc., and thought there are 3 distinct community types. And there was a lot of debate about it as well, and it took a few years to come to consensus how the gut microbiome looks like. So it's like a Swiss mountain landscape. This is density in the population, if you wish, and you have some peaks where lots of people fall into, and this is just purely genus composition. But you also have things in between, and for some of the peaks, you know, it depends how hard you look. You know, the ET1, ET3 could be, you know, if you have a more less rigorous look, could be just 1, and ET2 in that case could be divided into 3. You know, so that is still under debate. And the hope was that by time the big data will solve it. But it's unfortunately not the case. So it's just again back to these 3 enterotypes, and this is a very particular clustering method. Very particular distance, and also its genus level in a very particular taxonomy. So all these things actually play a role, and just go through one by one. So that was original stuff where we were excited about it. Then 7 years later, same method. This is sort of the comparable kind of thing here. Yeah, could be 3, could be even 4. And different people take it from there. And if you take other matrices, you have completely different answers to this. So that's another issue. All the methods have pros and cons, etc. And now we thought with 17,000 samples, so really clean samples, we know exactly, you know, only healthy, etc. We were very disappointed and went back to 2, but some people argued for it. So this is sort of, I think, inconsistent, and it probably continues. But if one goes underneath a bit, and that is, I think, in a metagenomics world these days, we have the standard classical NCBI taxonomy. I think it's still dominating, but a growing, fastly growing group of people use a new one, GB, which is much more consistent, more data-driven, and much more fine-grained as well. So, and if you apply not that taxonomy, but the other one, you are swapping again back to the 3 very quickly. And the reason is a genus, you have to lump species together, and the names change, the grouping changed, so the starting point changes a lot. So for us, it's basically, we think this is indeed much better than what we used to work on. So we switched a few years ago to this one. But again, things become more complicated. This is now the algorithms. Then it's a target function, the evaluation function. You take a CH index, a Gillett index, etc. and what kind of cluster. So at the moment, clustering, we favor the fuzzy clustering because it can model this landscape of the mountain I told you a bit better. But then again, what kind of statistics you apply? And this is not, at least a few years ago, we really asked specialized statistician. There's no good answer. So here, there is something that needs to be cleaned up. But again, we work with messy data on various fronts. So coming back to the confounders, it was just one discussion on one of these many confounders that might be there. When we compared all of them, given all the work for the last years, I think medication stood out as being the one of the known ones that really dominates the theme, but have in mind only 20% of the variation we can explain at that time. So it still might be something we haven't really on the radar yet. And I'd like to go back a bit into this. So we first found, again, that medication is a player, and now we can quantify it even. And how we came across this quite a few years ago, that was, again, typical association study. This was type 2 diabetics, and there was 2 Nature papers, fantastic correlation with the microbiome changes, looked all good. We had a third data set, also looked good. But in our data set, you know, half of the patients take metformin on average in high doses for a long time. And if you sort of dissect the metformin signal from a type 2 diabetes signal, then you see the strength of all that association was with the metformin and not with the disease. So what you see in black here, these are the typical curves. That was a total signal in black. In red is a metformin signal, and the disease signal is a pretty lousy one, actually. You still can use it here, but what is it good for type 2 diabetes? Maybe forensics or so, you know, if you analyze poo, you know somebody was diabetic, but you know, from a purpose, it was completed in a wrong direction. So we took this further, more recently on a large cohort, 2,000 people, and more than 2,000 people, and could see this is on cardiometabolic diseases and with lots of medication. Elderly mostly, again, medication is strongest signal, and indeed half of the association you would get with various markers. So here we had metagenomics, metatranscriptomics, we had blood metabolomics, all kinds of clinical readouts. Half of the naive association are confounded mostly by drugs. And indeed, the drugs that are subscribed in various subcategories, they override the kind of molecular markers. So if a certain molecule is increasing in disease state, the drugs make it push back on average. Problem here was, or also challenge, as I mentioned, heavily medicated people taking up to 8 or more than 8 drugs together, so polypharmacy, and in various combinations. So, and that means drugs can also infer each other, and indeed that is what we see. Good news is the combinatorial effects for the known recommended co-subscriptions work in terms of disease reduction. There are very specific effects, but we also see additive effects. So if you take a drug over a long time, so you have basically amplifying potential here. Take antibiotics, it's over 5 years, the number of courses. And the more courses you take, the gut microbiome richness really goes down, which is not good to an extent that some species disappear. And usually, you know, it's good to have them, or at least the healthy ones. And also the antibiotics resistance goes up. So with each course, you have selection pressure, you get something in. This is again over many people. But so at the end, and you don't lose them over time, which I still don't fully understand. But so it must be an advantage having them. But if you're ready for the next one and you're full of antibiotics resistance genes, it's also not so good. So, but we can even with these association studies see dose effects. It's back to the endotypes. You have 4 endotype model. And the more you take here, 2 examples, you really shift the microbiome in very different directions. So lots of things you can read out with the association studies these days because the cohorts get larger. But in the end, you have no mechanistic understanding and you have no causation. And so you need really other non-bioinformatics ways of get to this. And that brings me to the experiments in a way. So we have an EMBL nice setup developed over the years where we have lots of in vitro studies. We work with mice, all in a coordinated fashion. And they're cool stories. I just want to, this is coming up soon. It's James Hackett actually is leading it. And it's basically if male mice are under antibiotics while they mate, or in a mating time, there are quite horrible effects on an offspring. So they're via the microbiome. There might things that that we have to reconsider in our conceptual thinking. Again, this is just an anecdote. Point was here, different kind of experiment techniques. So this ex vivo, meaning you treat perturbed stool samples, for example, screen them by drug, etc. So you're getting closer to reality, because many people doubt whether mice models for microbiome are great. I think they're great in many respects, but not in all respects. So one has to see why make an experiment, and that is all lumped together with lots of bioinformatics expertise, really as a teamwork. I just want to go in a couple of these findings from in vitro studies, so amending the bioinformatics association studies. So the setup we have is basically robotics readout. Optical density is measured, and the baseline was quite from years ago that you could culture bacteria under defined conditions. And we have probably 200 by now. Then you can throw drugs at them. It's a 40 study. I come back to it. You can check for coordinations, for combinations of drug. And also, so far, this is drug versus bug, but also bacteria biotransform drugs. So it has also effects on various sites. And this is one by one, but at the end of the day, they live in communities. So we have now a new generation of defined communities where we can do similar experiment to dissect the individual contribution and then bug interaction effects. Just a few anecdotes out of this. This is a screen of 1,200 drugs against 40 commensals. They are here. I don't want to go into detail of the things, but the outcome was that more than 25 of the human target drug, cancer drug, etc., they have a side effect in a way that they at least deplete one of the bacteria in a gut of that community. So 40 was representative, but we have more than 1,000 in us, so it's an underestimate. So in each deletion of a particular bug can cause quite a few side effects, and indeed, we correlated with side effects. There's a correlation. But sometimes it's even the mode of action. The bacteria are involved also in the mode of action of the drug. So another thing then, if we move to synthetic communities, we stumbled across different challenges, and that is now the multi-omics part. So you try to see the effect by various kind of techniques of molecular readout. Metagenomics is one, metaproteomics, you have metabolomics here, you have metatranscriptomics. And they all have good insights. You have a defined readout for the first time. We still don't know a golden truth in our gut, for example, how it should be, how to interpret this. But each of them have biases, have limits. And it turned out that metatranscriptomic actually is a good central part, connecting to all the other things in the best way. So not the metagenomics. But again, that is a community translated to the gut. It might be still different. So very basic aspect that we don't know how good our tools or what they're missing is still not there yet. So we need, on that front, lots of systematic, defined approaches before we can go into the wild. So just a last anecdote on that part is so the surprises that come out. So again, I told you we set up a screen, a very small screen, 25 bucks versus 15 drugs, where we wanted to see what kind of biotransformations we do not know in that defined set. But the big surprise came up that a big fraction of the interactions between the drugs and the bacteria was basically what we call bioaccumulation. That means that the bugs swallow the drug, take it out of the pharmacokinetics, if you wish, and that's it. And so it was unexpected that we have huge, it's a new mechanism or interaction type between the bugs. It has to be factored in also in dosage concentrations, etc. And it's very individual. So again, and like this anecdote, we have in vivo, ex vivo, natural studies, association studies of interaction between bugs and drugs. But there's no one-stop shop of getting access to this. So this is a long-term project. It will take time. So we tried it because we have experience. We have a tool around called Stitch. This is more on the human side between compounds on protein interaction frameworks with side effect and other things. But this takes a lot of time because, again, like with the confounders, each of the individual components or confounders or methods has biases we don't understand fully yet. Okay, but that's just a few things where we are, dealing around in a gut part in collaboration with experimentalists. But experiments in the wild are much more difficult, but we can take that knowledge and have this in mind. So we built up, for example, cultivation facilities for communities now at EMBL in order to prepare for the upcoming wave we expect. Because this is an exploding field, particularly with climate change, all these societal challenges, looking at the world. being it the ocean, being it in the soils, agriculture, rivers, plastics, name it, antibiotics resistance even. There's big things that can be tackled if one has a database line for this. And that's what we're up to. I just want to go into the part I was promising in the title. But even the poo is useful. So what you see here is a kind of biogeography of the poo. These are the sample locations, even if it's only city resolution, etc. And you see at that level already there are different strains, there are different subspecies in different parts of the world. That's more geographic things, but now we can overlay with other types of data. So we have been for many years part of this so-called Tara Ocean Consortium. That's a ship, a sailing boat that goes around in the ocean, samples. And the first samples were 2009 to 13, I think. This focus on biodiversity. It was in dark blue. There was a kind of an Arctic cycle expedition afterwards. And this was from virus to everything that's smaller than 2 millimeters, even fish larvae, etc. So different organismal range. Lots of gradients, different depth layers, and in a standardized way, because it was missing, or still missing in that field. And actually, in terms of standard, Tara helped a lot because people adapted towards this kind of first kind of expedition. So there's still tons of papers coming out on different things, but here it's really baseline. So what drives the community composition in the wild? So, and what's different from the gut, for example? If you take this, and that's from the first expeditions here, these are the samples, temperature layer behind, and then also the regions, and the oceanographers classify these different regions, all defined or agreed on. And the question on what drives the composition in a different spot. So if you do by geography, here the color code, it's quite a mess, so just simple clustering here. But if you put against temperature, you have a fantastic correlation. I mean, for that field, fantastic correlation in a way. The plus minus 2-3 degrees, you can predict the temperature based on the community composition you have. It's a very expensive thermometer. Each sample was at that time 1,000 euros, etc. And you get it much cheaper with a camera, etc. But it gave what is the drive? It was temperature. Even that was much more complicated. Like in the gut with all the confounders, temperature correlates with CO2, with oxygen, with all kinds of other stuff. But because of the depth layer, we could sort of trick all these confounding issues and could nail it to temperature. Very basic stuff, but very useful to know. And again, human gut doesn't apply. We have cozy environment for our guts, unless we have fever or so. So it's rather constant. So other factors take over there, obviously. Okay, but this is sort of the biggest ecosystem, the ocean. But we can also go to the soils of the world. So we had, together with Estonian collaborators, they sampled actually across the world topsoils, and here we also see lots of novelty just in an explorative sense. And just go into one story, and that we have observed in a given tendency over many, many samples, we can correlate. So if there were more fungi in a sample than bacteria, they were kind of, the bacteria were reduced in diversity, and those who were there had lots of antibiotic resistant genes. So you can sort of hypothesize that these, the fungi shoot the weapons, and the resistance system in nature are selective to keep them alive there. So this the absolute latitude, and you see really if bacteria go up, fungi sort of are orthogonal in a way, if you wish. And interesting, that kind of anti-correlation, antibiotic resistant gene abundance can explain 50% of this. So it's a rather strong signal in that field. And you can now dream further what you can do with that information. Nice to know, but you can now pinpoint antibiotics producer. So if you over many samples, and here we need more, so we could, like the Basidiomycota are well known for correlating basically with antibiotic resistant genes. So they are present, lots of antibiotic resistant genes are present. But there are other taxa like the Mycota here, even sister groups of fungi, nobody has a clue what they're doing, but they must really be tough and produce lots of antibiotics. So that again needs follow-up experiments. You have to zoom in, you have to find them back, because in the ocean, there are currents, and what you see one day, the next day is gone. But there's huge potential, dreaming further with the data we have for practical applications here. But what you can also do, and is basically correlate different fields. So in a big world perspective, fields are very fragmented. So you have the oceanographers in the ocean, you have the gut guys, you have the soil guys, agriculture, they don't talk. So you just overlay their data. And so very rough, what you see if you do so circles here, you see that there are regional similarities. I just zoom into antibiotic-resistant genes again, big topic, how they spread, etc. And what you see not that you have regional patterns per habitat type, but you also have regional patterns that go from soil to water. And soil and water is really a very different lifestyle. There's not a single species which really thrives in both very well. There's overlap here and there, but not much. But the genes have spread, so how does it come? So that goes a bit more algorithmic, or not algorithmic, computational here. So you can, obviously the first hypothesis is must be kind of horizontal gene transfer involved, and there we have all these different mobile genetic elements, and we had talks about this here as well. And the problem was with that, each a mobile element, be it transposon, a plasmid, a phage, an integron, name it. They have different research communities, lots of nice tools, but very tailored to a particular application, and a nightmare to put them all together. So we thought, okay, step back, do it very simple, and try to catch them all in one go to be quantifying, how much antibiotic load, how many mobile element loads are there of any type, any known type. So the idea was use recombinases as a hallmark of any mobile element, but they come in so many different incarnations, homology wouldn't do the job. So so we use this only in high quality genomes, 84,000 high quality genomes, and classified this. And the second step was then estimating the borders of elements. So you have a seed, and that works very well. So how far is this element going? You have lots of nested elements, et cetera, et cetera. And here we used pangenomics, and that we could show that if you have 4 strains of a species, roughly, this is high quality, it's not max, then you have a very good border estimate already. So very generic for any element. And then the third step, you use knowledge-based stuff. For example, a phage needs lots of extra genes in that border in order to be a phage, and other stuff, what is an insertion element or so. And by doing so, you can really have a grand kind of survey of these mobile different element types. You have also cellular recombinase. You can also separate them. Conjugated elements here, phage-like elements. We have unknown stuff. You have no clue what it is yet. But bottom line is that transpositive elements, this is including transposons, they really draft the phages, at least the ones that are integrated into the genome. And the research community around phages is much bigger than the transposon community, because it was just not on the radar. So they also carry lots of antibiotic-resistant genes, so a perfect target if you want to go into a kind of area completely understudied. And we also figured that 70% of an average prokaryote genomes are covered by any of these mobile elements. It's a huge fraction for the total, and often these are completely under-annotated. So, and AIGs are really frequent cargo for good reason. So here again, this is a very naive computation approach to at least help us in these big surveys we are planning to. For transfer, you can now use these elements, any type, and see how they, within these 84,000 genomes, hop around. They hop around between archaea and bacteria, so really wide distances, long bits. And here, to be sure, we only looked for recent transfer. So we asked for a nucleotide entity more than 95%, but between at least families. So genus, family should be far lower in terms of, so you have a reasonable safe cutoff. That's much more potential. It's the tip of an iceberg. But even if you do this, it's a huge access of these randomly selected 84,000 completely sequenced genomes. They come from various environments, etc. So I expect a huge number if you get at some point really an overview of the globe. And that brings me to the overview of the globe. So what we have for quite a few years now collect, it's like stamp collection, all kinds of metagenomic samples from all kinds of habitats, put them together, annotate them, because annotation is usually not really consistent. And this is now a snapshot, I think, about 100,000 samples we have right now. This metadata, again, dominated by the gut, because lots of funding in the gut. But if you look at the genes that are in there, many samples, like the ocean samples in Tara, we sequence much deeper for all kinds of good reasons. So we have by now 28 billion genes in these samples. 16 are unique, and the clustering is still ongoing. I come to this. And we are in the process of, or have almost annotated all of them with various tools to be consistent also in analysis. And there are many research questions like the global regional gene fluxes. that can be applied now already with these data. Although it's a huge amount of data, and again, storage issues, CPU issues, all that stuff hits very hard now because the field is exploding, being in agriculture, being in the ocean, in the coast, and lots of data come in. So it's a gap now between raw data production of any kind of consortium or group and ending up somewhere in EBI or other resources. Just give you, from a algorithmic challenge point of view, you know this, so we have different pipelines in order to analyze the Metagenomics data. They have to be assembled, binned, integrated with known genomes, genome information, and that quality controlled in voice. And if you just go into the MAC business, so metagenomically assembled genomes, like in a human field, it's a complex pipeline how you do it, and everybody thinks he can do or she can do it better than the other. There are lots of variation, and it's really a challenge now because we have now probably 50, 60 groups at that scale doing these things. Everybody has a pipeline like this. Every pipeline looks different. Every parameter set of a program is different. And there are good reasons to argue for any of these pipelines. That leads now to a really big mess, not only in naming. So even if one sample, you have 50 different interpretations. It's not one genome, it's thousands of genomes. The abundance, the genes, everything differs a lot. And for an outsider coming in, it's pretty hard to figure out what to take and why. So this is sort of a community effort, in fact, to at least have an idea. I mean, there's no way of agreeing, but at least an idea how to liaise and come together to present it for scientific purposes. This is sort of another layer is the annotation of these things. I mentioned the different taxonomies already. There are lots of functional annotation tools, and many of them have error rates for good reasons, but it's hard to improve. There is improvement there going on, and we waste a lot of energy, and it's not good for the climate. All these computations we have to do on these fronts, storage limits. It's I don't want to go into this. I mean, we almost can keep up with the human front here, and genome front. And again, the pipelines, they evolve, the tools evolve, improve. So every year you have to redo it. But then, how many update cycles you can afford? So all these things that everybody knows already, but you can do science with it. So this is just a survey on an old data set that was frozen in 2017, based on 2.3 billion genes. Again, 28 we have now from 18 different habitat types. And again, I point to the 2017, and the paper came out 2022. It's a long time in an exponentially growing field, also data-wise growing. So what we did here, we sort of by habitat type, we just mapped the genes into habitats, and then we see the number of detection of those genes at different kind of cutoffs, so 95%, because people also do not agree what a good cutoff is here. This is, we think, is about species level. So you have one gene, one ortholog from a species, and I think in protein database they clustered 90%, but we can down with sophisticated homology searches to roughly 20% plus statistics behind us, so a really wide family definition. So in all these different layers, what you see in almost power law, the number of detection and the number of genes in a way, and that is, in evolutionary terms, it sort of indicates that most of the genes might have a nearly neutral evolution. And that's true for most genes of genes. That is completely counterintuitive. If you take an E. coli genome, you have 70% of genes annotated as having cool function, implication, the function is selected. And you might have 3, 4 genes in a genome you have no clue about, and they might come and go. But it fits nicely with the pangenome, or with the open pangenome idea. So the more sequence, the number of genes still goes up, because each genome brings 1 or 2 little genes that come and go. But if you have then trillions and whatever of these things, if 70% is always the same, they're conserved, but all the rest is sort of adding up, you indeed come to that kind of view. So that's a big claim. You have to be proving it. And one way of proving is we looked at conservation of gene neighbors. So if they're in operons, there should be a selection on this. And you could see a tendency here. So number of detections, again, plotted here. We can look for PNPS in a sample, so positive selection per sample. And for E. coli, we could even, so we mapped the wild E. coli with the things we had. the plant genome we had from quality ones. You can even go per position and indeed see selection is correlating with abundance. So that implies that we might be able to sequence all the species on Earth, but not all the functions. They come and go, and you would never see it. It's like Walter Fitch many years ago. He tried to figure out viruses in the US, and when he went around, the virus had evolved already. He could start again. Things, they are so dynamic in a way. Okay, so but you can do now other cool things using very basic tools that got almost forgotten for many years, HMMs, and look like diagnostic in human bug. You develop biomarkers or bioindicators, it's called, in an environment for certain processes. Take carbon fixation. Everybody bets on the Calvin-Benson-Wasem cycle, which is well studied, should take all of it, but there are many other cycles that have been described in one bug, and we can project it again back to the whole habitat. So again, this is now a bit more forward-looking, but again, the problem for this is really consistent structured metadata. So, and this is, for me now, biggest practical challenge, that we go now really into the wild. And that we do go in the wild, I'd like to explain you with a project that has started, but it's just a dream so far. It was more than a dream, not ended. And it's a big EMBL project with lots of partners on traversing European coastlines. An expedition, and EMBL is not into expeditions, molecular biology after all, for really bringing the molecules to the ecosystem. And the main expedition will be in 23 and 24, but we did pilots for Corona already. Then unfortunately we had to stop. We did one in Roskop, had a 1 month warm-up in Iceland, so not just one site, but a few sites, and testing all the logistics. And the idea is really going with Tara, the boat. We rented it, let's say, for the 2 years, and with a fleet, it's I think in the order of 4 different vehicles with different purposes, to go along the coastlines and collect with a highly standardized metadata, like metals, like toxins, environment. We have a library of 1,000 environmental toxins at each site, etc. So whatever our knowledge is, put it together into this one big expedition. And then coastal region, I indicated already, because it's 2 very different ecosystems, still genes hop. Why? And it's affected by, has high biodiversity. We expect also lots of biodiversity stuff, but ocean warming, acidification also, but also by lots of stressors. So rivers going in, and it's getting dangerous if the mafia puts some rubbish in, and we are passing by and having statements. So it's also, we have to check very carefully the legal requirements, but this is underway already. Okay, just the idea is having it together with some partners like EMBL is a service infrastructure of Europe on marine stations, so have some baselines here, and doing really with a wide range of different collaborators with different kind of backgrounds. So and again, the idea is really open science, so the data will come out after quality control. And we have lots of plug-in projects where people use the infrastructure and can put their own projects in. Just to explain, it's only 120 sites across here. They're sharp gradients, so local ones, but also the wide ones. We explain temperature, obviously, from the north to the south. And I mentioned already the stops. That's not so relevant here, but it's a complex logistics because the samples have to be shipped, et cetera, et cetera. And but I'll show what kind of idea on the sampling is, and that we tested already. So there will be transects. So with the boat here, that comes by Tara offshore, and then transects a few hundred meters inland, where things are not so affected by water, where it's a mixing zone, particularly if you have tides, water comes in, retreats, and seeing where that's a hot spot of exchange, or is it aerosols, because dust clouds or whatever, that make functions fly, if you wish. And but we also go into the sediment, and here the ambition is not only go from virus to fish larvae, but really go to animals. We have a number of model animals, like worms with their guts, microbes, that we, population-wise also want to study here. This was a thing where we have the budget for, but now we expand. As I'm speaking, we try to get more people hooked into this, and this is sort of on top of what I said before. These are things that we got in via VORUS, what we call plug-in projects. One was a big one. It's a new grant, also in coastal monitoring. So we have now really probably sediment stuff is we have acoustics. This is more for fish populations. What we can see, can be correlated to sediment stuff and other parameters, etc. And all kinds of other measures. There's 3 core projects. I mentioned the worms. There's a big project on symbiosis, mostly imaging driven here, because some eukaryotes manage to get the cyanobacteria in and make them grow, make them fix more carbon than in the wild. So mechanistic stuff. And I will of course talk a little about this stuff. There's not much out yet, so that's all ongoing, but we have now data in from Naples, from the first pilot. And this is just again as background. And questions are really, what is the biodiversity to start with? Is there anything in any site? And what's the impact of chemical pollution? Is there the dispersal of molecular functions? Is this mobile genetic element, or is this just a bug dies, DNA is out, and somebody nibbles after degradation? So very naive, but the data will tell us. So we had already very interesting observations in the first, again, long introduction for one slide to come on the results now. But so let's, the first thing is Naples, the Gulf of Naples. You should know it's one of the, most polluted river goes into the Gulf, far away, but there are also pristine sites and mixed sites near the harbor, etc. And there is volcanic soil around it, that's Naples, and there's lots of chemical pollution as well. So I don't go into the pollution, but just on a soil thing, on a soil, and generally, indeed, we see almost no species by our definition that hops from water and soil. But we see an unexpected large archaebacterial biodiversity. We discovered novel orders of archaebacteria, all kinds of stuff. And we think they're needed to really make the nutrient-poor volcanic soil accessible to bacteria. So in the beginning of a kind of processing chain. And but that is just on a structure, how the community look like, very different from other sites we have seen so far on coasts. But also these soil bugs are vastly enriched in sulfur cycle, which makes sense for the environment, but we see lots of novel sulfur processing enzymes. We only have the operons, we know they belong to this, but we have no clue substrate-wide what the biochemistry is. But it's a huge novelty just on a sulfur metabolism here, just one site. Okay, so with that, I can just don't give a summary, but just reflect again on the challenges I see of various sorts. And for example, basic algorithmic, there's this Kami challenge for the metagenomics assembly. You see movement, so things do improve, means there's room for more improvement. It goes up quite far. That's on assembly binning, taxonomic profiling, etc. I alluded to the clustering again, because several methods that work pretty well with a few samples we had 10 years ago, they now completely fail. We think it's because of certain heterogeneity issues and biases, but we don't know yet. But they don't, very promising ones, they don't work, at least in our hands, on these big data sets. We explore this right now. So there's, again, more here. I mentioned already the annotation transfer. We talk about billions of genes. How do we consistently take it over and avoid errors being taken over? Particular antibiotics resistance is known to be error-prone, the carryover, because there's a relationship also to normal metabolism, normal mechanistic cell defense, and how do you distinguish? And there's a huge, inflation of antibiotics resistance genes. So if one knows what the shortcomings are, it's fine, but you can make conclusions far too early based on those things. I just touched about the cloud, not on the cloud, but the computing stuff. So we have to use a cloud for many of these computations. You have a German research cloud, it's very convenient. You can access, it needs your tools need to be cloud ready, obviously. And I alluded quickly to the data integration issues, and then you have to see, this is a human gut very prominent, you have these study biases. So same protocol applied in two different labs, same kind of set, we did these tests, very different outcomes in the processing pipeline. There's so many stochastics, it takes from a stool taking to the freezing, whatever. So this is stochastics one has to live with. It's very hard to control very tightly, but it leads to huge biases and you have to sort of compensate for later on. You have the issue of bridging spatial, mostly spatial scales. So from the molecule to subcellular level to organismal level, also organismal scale, from virus to whale. So it's all in the portfolio, it belongs together somehow. And there's lots of conceptual stuff. I mentioned biases already, the confounders, the batch effect, the study effects. but also technical artifacts that you can't avoid, be it in the methods, but be it in the sample processing. Lots of stochasticity, so particularly human gut microbiome, if we sample in the morning and the evening, it's different. The one day after, depending what you have eaten. So one has to live with this and or factor in. In a wild, there's lots of seasonality, actually even on the human side. In winter, we are quite a bit different microbiome, right? But here it's lots of statements in the cross-sectional study, as I mentioned, they're very hypothetical because you have to see the whole year how a site evolves over here. So we have lots of time series built there as well, and missing data without end. So just a thumb, I had 5 minutes to think about it, and I'm sure there are many other things that affect. Some are computational, other are more conceptual. But with that, I want to leave it here and can only encourage everybody, this coming world, particularly with the climate change, the new generation, much more interested, the donors. So we have lots of philanthropic support for the TRACK project. So usually the old man and the prostata, there's clinics these days, my kids and the future of the planet. So many of these are smart guys. There's a shift of funding now in the direction. I'm rather optimistic that much more is to come. And with that optimistic outview on the science of the planet, I like to acknowledge quite a few people. First, of course, the research group. This is the, before COVID, a nice team, different nationality, different cultures, different skill sets. Lots of alumni, lots of visitors. Well, we have fantastic collaborations on the tools I mentioned internationally, really long-term collaborations, but very open still. And there are lots of consortia on a human gut, but also coming now on what we call planetary biology. Tara Oceans, for I think 15 years now we work in that framework. It's really fantastic. And more to come. And again, you could see my excitement over the STRAC expedition, which hasn't even finished yet. So I hope we don't screw up in the data processing, but so far it looks good. Thank you very much for your attention. And maybe my last statement is really, as a bioinformatician, I can live in allergies. So I see the human gut, and there we go for diagnostics and for individualized treatment. But in an environment, it's the same. They call it not biomarkers, but bioindicators. And it's not treatment, but again, there's various dedicated regional bioremediation needs, and the microbes might play a role there. But for that, like in a human, for therapy, we need more mechanistic understanding, but we are completely short in the wild. Thank you very much.
So we'll open that up to questions for Pierre. While people gather their questions, I do have one for you, which is kind of more speculative. But if you had advice for the graduate students who are attending genome informatics, what would your piece of advice, thinking forward, you give us a lot to think about here, but what would be your one piece of advice to graduate students?
Well, it's different. So you have to go with the fields that are growing, and I mentioned one growth field for sure. So you have lots of career options in that sense, and you have to team up these days. This is an important thing. The networking, a conference is one way, because so many things, so many tools around, so many things, so many concepts you have to put together, and a single person, even a brilliant one, cannot handle it. So networking is another big one in the field, and leave the others do, and don't all do it all yourself. Take your bit, but connect to the others.
Thanks. Really interesting to think about. There's a question over
here. Wonderful talk, Pierre. Thank you. The Coastline Project's super exciting. In National Geographic this month, there's these incredible images of microbiome from the Black Forest floor. And I was just wondering if you had done any sort of matching of all the sequencing data with imaging, a bit like spatial transcriptomics or something.
Yeah, so it's another big area with future. It's sort of the multi-omics combining with imaging technology. So we are very much on this stuff. Actually, some of the projects involve lots of imaging. I didn't touch it because it was on metagenomics in a sense. But the same stuff comes in the imaging world, EM, electron microscopy in particular. There are still no really proper standards, how to communicate the data. The database is out there, it's getting there, and EMBL, particularly EBI as well, they push quite a bit for this. And so we work on a cute bug called mycoplasma pneumoniae, so with other collaborators, and ideas are really, you image, you burn the cell, but still the genes are still there, so you can see the cell state after being frozen, etc. So there are lots of things, and I think it's another very future area to bring this in. At a large scale, for track, for example, we still fight with the permits. We intend to have drones and to characterize basically usually ecologists say count the plants and the ants and whatever, but trying to be sophisticated there. But I have so many dimensions of that project and has to prioritize. Here we have issues a bit with legal permissions because this is tricky ground, but would be great. So I think it's a future area. We have some plans, but I can't give you anything concrete, maybe some anecdotes, but it has to come and people are aware of it.
Interesting, thank you. We're going to take one of the online questions.
So you showed that many of the drugs impact at least one of the bacteria in the human gut microbiome. Are there common features or properties of these drugs that...
Can you repeat this, please?
Are there common features that correlate with a higher or lower effect on particular bacteria, and can they be used to engineer better drugs?
Yeah, no, it's a range. It's a range, but yes, on the last one, so it's a range. Some of the drugs we observed, it was not known, some aromatic drugs, they were like full broad spectrum antibiotics. They killed everything. It was against rheumata. So others very specifically took only one of the 40 bugs out in that study. So it's a range, but the idea is indeed repurposing. But again, our limited knowledge, I didn't touch it. We did the drug combination. So we thought if you have two specialized antibiotics or drugs in that respect, killing different bugs, if you put them together, you get that all. You have a broader range. What was happened in these tests, we had a very specific range putting two drugs together. So there are explanations for this, but we are really pretty much in the dark predicting effects of stuff. But indeed, like taking that one drug I mentioned, which is full-spec antibiotic, but it's marketed against the human disease, there is potential to adapt those. So there are thoughts, but there's no company yet spin-off going practically about it.
Interesting. All right.
Hi, thanks for the talk. So, I mean, you showed some associations between various conditions in the gut microbiome, but I imagine, depending on the condition, the arrow and causality might point in the one or other direction. So do you have some insights into how you can tease that apart?
Yeah, it's a whole research field coming from association, directionality, etc. So for the environment, some things are at least easy. So if it rains and you see a change afterwards, you can assume it's probably the rain, not the bugs influencing the rain. But for many other things, it's obviously very tricky. So even from in a human part, if you assume you take a drug, something changes, you would think the drug is causing it. But it can be indirect. The change you see might be caused to some. So yes, we do it. There's actually clever mathematicians trying to, and there are methods out for this. But depending on the model you apply, so we used a few of those. There's a certain modeling framework for getting directionality in. It's therefore I showed the endotypes. Nothing is black and white, so you can get different answer depending on your assumption you build in, and some you do have to build in. So it's another. Who wants to go? This is a field not only in a human gut, but in the environment even more. Because in the environment, there are many more factors than we witness in a human gut. And there are plenty of them already there.
Interesting. Right, are there any other questions for Per? Yeah.
Thanks for the great talk. You mentioned all the differences between the bioinformatic pipelines and how that can create some confusions. Is there any attempts for sort of standardization or more cooperation so that sort of there can be more standardization of these tools? Do you feel that's needed?
Yeah, it's a bit of my feeling is it's a bit of simulation as somebody described in the morning or so. Yes, there are attempts to standard, but at the end, nobody wants to change. So because it's a huge effort, particularly if you look from a research group. You have your students, your postdocs coming, you have a certain time frame for doing it, you have a turnover. It's hard to change your things completely or so. In some areas will be successful. And also, I mean, with standards, I think communication is a good starter, and that happens. And there are standards. I think EBI drives a few papers on basic minimal annotation standards. In some areas, you can do it. In others, particularly on the tools, they evolve very quickly still in the field. So if you zoom in too quickly, you might lose out the next step. So it's not a black and white. Yeah, standards is great, but you have to see when can you apply it, where it makes sense, and where you find a consensus. For the endotypes, we fought for 5 years to get sort of a conception consensus on how things look like. So I think they come, and also this CAMI competition helps a lot, at least rationalize the power of the drug, because if you publish a paper, obviously you're always the best in certain angle. So it's very hard for which problem, which tools are better or not. For data sets, it's perhaps easier, but I don't see it so far. It's more the strongest is winning kind of attitude, which might also help, but we'll see. Yes, attempts are there, discussions are in various forums, but little progress because of the complexity.
Great, well, please join me in thanking Pierre for a phenomenal keynote talk.